Okay, good morning, everybody. My name is Michael Freely, and I am an assistant professor in precision medicine in the School of Biotechnology here in DCU. I am also the program chair of our highly successful MSc in Diagnostics and Precision Medicine, which has been running over the past 5 years. So today I'd like to give you an overview of what this course entails. It is a hybrid learning program that details how the latest advances in both science, technology, and medicine are enabling diseases and conditions to be diagnosed more accurately, particularly through molecular analysis rather than through diagnosis of patient symptoms. With that, the course also details how treatments can be approached more individually. As a whole, diseases and conditions tend to be grouped as a whole, and people tend to be treated with a one-size-fits-all approach. And we know very clearly at this stage that does not work for most patients. A good example would be chemotherapy, working for some patients, but not working for a lot of patients with apparently the same disease, for example, breast cancer. So with the better analysis of diseases through molecular techniques, we can now pinpoint the molecular basis of disease and treat those diseases more effectively, resulting in improved longevity and even cures. In addition, advances in both science, technology, and medicine have enabled even the identification of those at risk of developing a disease or condition before it even occurs. And typically with medicine, we would, treat patients once the condition has occurred. But identifying those at risk of developing a disease or condition allows you to perhaps do something before this disease manifests. And of course, prevention is always better than cure. So in a nutshell, that is what our course entails. It's been running for the last 5 years very, very successfully, and a huge number of our graduates have gone on to have very exciting careers, both in the science industry, both the biopharma, the diagnostics, and indeed the medical and healthcare industries. Our course is offered on a 1-year full-time basis, that is almost for 11 months, or you can take the course up to 4 years part-time. So you can do the course part-time over 2, 3, or 4 years, and that's where the flexibility in our course lies. Over the next 12 or 13 minutes, I'd like to give you an overview of what precision medicine is, then go through our course structure in terms of how we deliver the program. I mentioned it's a hybrid delivery program. I'll go through the modules and also how we assess you and your learning, how we assess your learning, how you're doing throughout the year, give you some information how to apply. And then, of course, a lot of applicants are always interested in, what careers lie ahead for each student. And we track all of our graduates, and we can see the very exciting careers that they've ended up in. And finally, if there's time permitting, we look at some of the testimonials, but these are also available on our course website. Okay, so first an overview of precision medicine. So really what precision medicine is being able to treat patients with the right medicine at the right time with, and really all of this has come about basically through advances in our understanding of what is a normal state and what is a disease state. And this has been, led by molecular biology revolution, because with that now we can pinpoint the molecular basis of cancer, we can pinpoint the molecular basis of inflammatory diseases, but also inherited diseases which are passed down from parents to offspring. And really we've come a very, very long way, because Hippocrates, who is widely regarded as the founding father of modern medicine, he believed back in the BC era that, for example, cancer was caused by an imbalance in certain liquids in the body. For example, he believed, and he was mistaken in this, of course, that the body was made up of 4 different hues or 4 different liquids, such as phlegm, blood, black bile, and yellow bile. And he believed, and we have no proof of this, but he believed that cancer was caused by an excess of this liquid called black bile. We know that that's completely not the case now. We know that cancer is a disease of the genome, and that genetic aberrations in your DNA code can lead to altered cell proliferation, where your normal cells turn into cancerous cells and proliferate too much. We can now pinpoint that in the modern molecular era. So all of these advances in molecular and cellular biology have allowed us to pinpoint the molecular basis of disease and look at, for example, molecules like nucleic acids and proteins, and pinpoint where someone's cancer, what are the drivers of a person's cancer. And so what precision medicine really is taking into account both prevention and treatment strategies that takes each patient as an individual into account. And that is looking at people's genes, the environment that they're in, and also their lifestyles. And with that information, coming up with the right medicine at the right time for the right patient. There's no point treating a patient with the wrong medicine because they're not going to benefit from it. And we know that every drug has a side effect. Some of these drugs have mild side effects. A lot of drugs have very serious side effects. So it's important that if a patient is going to be administered a drug, that they're going to benefit from the drug and not experience all of these, terrible side effects which can occur for patients who actually don't experience any benefit. So we want to avoid that. So really the goal of precision medicine is to integrate the latest advances in science with the latest advances in technology, for example, diagnostics, and being able to pinpoint the molecular basis of disease, and to marry those two technologies and disciplines with medicine and apply science and technology to medicine with the goal of delivering the right therapy. Here's the dart, delivering the right therapy to the right patient at the right time and hitting the bullseye every time so that every patient can benefit from the therapy that they're being administered. And this has, shifted away from the one-size-fits-all approach, which has traditionally been done. For example, we would see breast cancer patients as not being a single disease. Breast cancer is a heterogeneous number of diseases, and this has only been brought about by advances in molecular medicine and diagnostics, whereby we can stratify patients, whereby the breast cancer looks the same, down a microscope the cells look the same, and that they're tumours. But when you analyze the molecular basis of their disease, we can stratify or we can segregate breast cancer into a number of different subtypes, and they're treated completely differently from one another through these advances in science, technology, and medicine. For example, we now have hormone receptor positive breast cancer. We have HER2 amplified breast cancer, which occurs in about 15% of all breast cancer cases. And thirdly, we have what is known as triple negative breast cancers, which don't express these markers such as HER2 or the hormone receptors on their surface. And when patients with these subtypes of breast cancer are treated, according to the molecular stratification here, they're treated with completely different medicines, which have been shown to give them the best outcome and to control their disease better. And a key to implementing precision medicine is the diagnostics, understanding that what are the molecular aberrations in this cohort of breast cancer patients that all look the same down the microscope, but when you apply the molecular diagnostics assays to those breast cancer patients, you'll see that they will segregate into different subtypes, and then they're treated accordingly according to the molecular subtypes of breast cancer there. Another example is lung cancer. Traditionally, lung cancer was divided up into small cell lung cancer or non-small cell lung cancer by a pathologist looking at those lung cancer cells down a microscope and seeing whether they were small cells or large cells. We've looked at the molecular analysis of lung cancer now, for example, particularly looking at non-small cell lung cancer, we see a number of different subtypes appearing, whereby a cohort of patients that all look like they have the same lung cancer have vastly different molecular wiring of their cancers, such as EGFR mutations, ROS1 translocations, BRAF mutations. And these are only a small percentage of the total cohort of lung cancer patients. And for these subcohorts of patients, we now have excellent drugs that can target these molecular aberrations and give these patients the best chance of controlling their tumours. And these drugs have been shown to be much better than, for example, the one-size-fits-all approach, such as chemotherapy, because they've been shown in clinical trials to extend patient survival. Now, at the moment, unfortunately, these are restricted mainly to lung cancer patients whose tumours have progressed. And there's a good reason for that, and that's because these patients need these therapies most. But the beauty of precision medicine is that we're applying it to earlier stages of diseases when the tumour is still localized in the originating tissue. And this is resulting in actual cures, which is fantastic news. So the goal really of precision medicine is to target the aberrant pathway in that person's tumour. And rather than looking at the whole cohort of different patients with apparently the same disease, we're treating individual patients. And this is resulting in a tailored approach and an alternative strategy to the one-size-fits-all, and indeed the trial and error approach, where a patient gets a therapy. Let's have a look and see, does it work? If it doesn't work, let's try another therapy. If that doesn't work, let's try another therapy again. And this trial and error approach doesn't work because the patient gets all of these horrible side effects from a drug that is not working for them. And so we're trying to move away from that to use those darts and hit the bullseye every time. This has been proven to result in increased patient survival. And indeed, these targeted therapies, which I showed you, for example, for non-small cell lung cancer, have indeed replaced chemotherapy for many types of diseases, both in lung cancer and in breast cancer and other diseases such as melanoma. So, of course, precision medicine has it all sorted. Is that right? Absolutely not. We need the best minds to be recruited into precision medicine to help us, extend precision medicine to not just cancer, but to inflammatory diseases and inherited diseases as well. An example of this is where we are with an inflammatory disease. This is a patient, a young boy, 18 years of age, with psoriasis, which is an inflammatory condition of the skin. You can see these red patches here. This patient was treated with the standard anti-TNF therapy. These are antibodies that mop up inflammation and work for a lot of patients with psoriasis. You can see after 3 months that this patient did not respond to the standard anti-TNF therapy. And there was no reason why they went to an anti-TNF therapy other than the fact that it's known to work for most patients. Didn't help this young guy, unfortunately. However, the patient was then treated with a new FDA-approved drug, which was shown to completely resolve their psoriasis. It targets a different component of the inflammatory cascade, not the TNF arm, but it actually targets T cells. And it shows in this patient then that there's actually T cells are driving this patient's disease, because when you block those T cells with this drug, you can resolve this inflammation. So you can see the one-size-fits-all approach does not work for all patients with inflammatory diseases. So we need to do better at diseases like this, because at the moment, patients are given anti-TNF therapy up front with the hope that they'll work, but we know they don't work for a lot of patients. So like cancer, we need to apply molecular diagnostics and molecular analysis to these types of diseases and show, for example, T cells and immune cells driving subsets of diseases here. And that's why we need the brightest minds, both from science and technology and indeed medicine, all coming together to bring precision medicine to the masses, and to really improve patient outcomes and indeed healthcare. So quickly, I'm going to go to the course structure. Our course is primarily delivered by DCU, but we have a number of modules delivered by our partners at Arizona State University through the International School of Biomedical Diagnostics. I mentioned that our program is delivered via a hybrid learning approach, which enables you to study at a place and a time that suits you. A lot of our lectures and modules are delivered from the classroom in DCU. There's a picture of me delivering a lecture on multiple sclerosis. We record all of our lectures from DCU and put those up online on our online learning platform called Loop for you to review at any stage during your time with us. That allows you to have access to your slides and your recordings and helps you to revise your material. Our lectures from Arizona State University are all delivered online as pre-recorded lectures, and we support those at DCU with live tutorials, which are also recorded from the classroom. Just quickly, because of time, I'm just going to go through semester 1 modules. The first module is Introduction to Cell Biology and Biotechnology. Here you look at all the advances in cellular and molecular biology and how we can make drugs and therapies from living cells. Recombinant DNA technology looks at how we can manipulate both the cells that make these drugs and indeed manipulate the genetic sequences encoding these biological drugs to give us better drugs. We also deliver a module on fundamental and applied immunology, so how our immune system is important for targeting pathogens, but how we can manipulate our immune system, for example, to target diseases such as cancer and to make better vaccines. Molecular Diagnostics is delivered by our partners at ASU, and here you learn about all the key technologies whereby we can pinpoint the molecular basis of disease. And this all culminates together with the cornerstone of our semester 1 module called Precision Medicine 1, where you will learn about how these molecular diagnostics and targeted therapies are coming together. So how the science and the technology is coming together and being applied to medicine so that we can deliver the right medicine to the right patients at the right time. And we see some great examples of through breast cancer, melanoma, colorectal cancer, chronic myeloid leukemia are now being treated differently than the way they were previously. In semester 2, we have 2 modules from ASU, Translational Bioinformatics and Applied Biostatistics, which are delivered from ASU by our partners there, but are supported with tutorials at DCU. And again, these are all recorded. We have a module called Laboratory Techniques in Biomedical Diagnostics. Here you learn about all of the laboratory skills that are needed for a successful career in precision medicine. Then we have a module called Professional Skills for Scientists, which looks at all the soft skills needed, for example, science writing, science communication, the ethics of precision medicine, research integrity when you go into a lab, making sure that you record your data properly and have everything doing things right by the book and avoiding any of the previous mistakes where science has fudged results, for example, like that. We want to avoid that. This all culminates in Precision Medicine 2, whereby we look at how precision medicine is being applied quite slowly and needs to be applied more, and certainly in more emphasis on other diseases other than cancer, such as inflammatory diseases and inherited diseases. So it's one of the most exciting areas of precision medicine, where precision medicine is actually going in the future. In semester 3 then, usually the end of April or the beginning of May, you will undertake a literature review, a research project, and a presentation. This is worth 30 credits, which is worth 1/3 of the whole course. Typically, this is undertaken in a DCU laboratory full-time for 12 weeks. So you go into a designated laboratory and undertake a full-time project there, working on cancer, working on inflammatory diseases, working on inherited diseases. If you're working in industry, our course is flexible. You may take your project in your place of work, subject to some terms and conditions. The research project is broken down into a literature review, the project itself over the 12 weeks, which you write up as a dissertation, and then you give a presentation to the principal investigators on our master's program. Over the last number of years, we've introduced an alternative stream to the research project, and these are called masterclasses in precision medicine. And here you will undertake a number of short laboratory courses in precision medicine, and you'll get to treat 2 melanoma cells with an FDA-approved drug, which is used to treat patients both in Dublin and all around the world to target a particular molecular aberration. And you'll see that one melanoma cell line responds to the drug, the other one doesn't, and this reflects the clinical situation where a person with this mutation in their melanoma tumour benefits from this drug. And we'll do all of these techniques shown here, which I won't go into today. And with that then as well, you'll also get to write that up as a research manuscript. We'll give you the skills in that. And you'll also prepare a grant proposal, which is a key skill for a career as a research scientist. And finally, you do a presentation. So there's 2 alternatives here: the normal research project or the master classes. How do we assess you? It's 100% continuous assessment. As an example, here is the 12 weeks over semester 1. You'll see at certain weeks along the course, there are assignments or there are online exams or in-class tests that you must attend. And these are used to assess your learning over the course of both semester 1 and semester 2. So we don't use formal end-of-term exams in an exam hall to assess your learning. So you can plan when your assessments are due throughout the semesters. How to apply? You can find information on our course website. Just Google the MSc in Diagnostics and Precision Medicine at DCU. You'll be taken to this link, and from that, then you can learn how to apply. Our course requirements: we require a second-class honours degree, so a 2-1 in either life sciences, a chemical sciences, a pharmaceutical sciences, or medical sciences. Alternatively, we accept a person with a second-class honours lower level at 2-2 if you have relevant industrial experience. Career prospects: many of our graduates have gone on to have highly successful careers in both the biopharma industry and the pharma industry, such as Pfizer, Novartis, Bristol Myers Squibb, and Gilead. They are a company who make CAR T cells. They're personalized therapies where they genetically engineer your own T cells and give them back to you to fight cancer. Some of our graduates have gone there. Some of our graduates have ended up in the diagnostics industry, such as Leica Biosystems and indeed hospital laboratories. Some graduates have ended up in life science companies, such as AccuScience, charity organisations as researchers there, such as the Cancer Clinical Trust research. And also many graduates have gone on to further research careers, such as undertaking a PhD, either in DCU or in other institutions such as TCD. Some of these are linked with industry as well. And finally, some of our graduates have gone on to do medicine, go back to do medicine after this, and they found the precision medicine course very, very beneficial to them. So testimonials, I know I've gone over time here now. These testimonials are on our website, so please have a look at those, and you'll see that all of our graduates really enjoyed the course and have flourished since they've left us. So with that, I'd like to take any of your questions and look forward to speaking to you in due course.